In this lab, we're going to be testing the sensitivity of microorganisms to antibiotics, common household cleaning agents, and UV light. So I'm going to show you here. You can see a stack of TSA plates, 1 and 2, two cultures, Salmonella typhi and Staphylococcus aureus, plunger with 8 antibiotics, tweezers, and common household cleaning agents. Here on the left, you can see a lamp. That's a UV lamp. And here on the right, this one, here, that device is an incinerator, which will help me to sterilize the wire loops. Next to it is the Bunsen burner, which I may also run for certain reasons, but we'll see how it works. Anyway, you've got it. So, having said that, let's make sure that everything is clear, and we're going to start pretty soon. So I will inoculate my plates first. I'm going to put this stuff away for a second, and I'm going to take a wire loop, you can see it, and I'm going to sterilize it in the incinerator. Incinerator is on my side, you can't really see it, and that is not super exciting. So the wire loop is indeed incinerated. I'm going to let it cool down a little. And I think that's sufficient amount of time for it. So I open my Staph aureus sample. Stick the wire loop in. And I inoculate my first plate. So let me explain what's going on here. I'm trying to create a really thorough bacterial lawn. So I am inoculating all the nooks and crevices of that plate. Trying to make sure that every square millimeter or inch, if it's an imperial system, is covered with bacteria. So I sterilize. In the meantime, I have another loop ready. So I'm going to inoculate a second plate. In the same exact way. So you see, I try to use, I use separate loop for each inoculation to mitigate potential for contaminants. Okay. So it's inoculated. Nicely. And densely. So that's number 2. And we're going to do number 3 now, sterilizing my second loop. we are basically done with Staph aureus. Again, nice thorough inoculation. Okay, excellent. Staph aureus is done. And our next inoculation is salmonella. It's not going to be different, so you're not going to watch how I inoculate salmonella plates. We have inoculated both of them, and now first we're going to apply antibiotics. Looks pretty cool. It's a plunger for so-called Kirby-Bauer diffusion test. 1, 2. This thing is done. Next is going to be, unfortunately, pretty long. I'm going to show you only on one what I'm going to be doing. So I've got some sterile tweezers here. Okay. Move this wrap away. Got a few sterile tweezers. Okay. And I'm going to bring some sterile paper circles right here, okay? And I've got my common household reagents. So before I start, I'm going to label what I'm going to be doing here. So I'm going to have Lysol, I'm going to have hydrogen peroxide, I'm going to have scope, I'm going to have microquat, I'm going to have ethanol, and I'm going to have silver nitrate. So I'm going to place the circles over the corresponding labels. So what's the first? First is silver nitrate. So I'm getting a tiny little paper circle. Put it in there, okay? Make sure that it has plenty of substance on it. And even maybe, drip some stuff off. Place it here, okay? I'm going to make sure that tips of my tweezers are sterile. Put out a silver nitrate. Next is hydrogen peroxide. Okay. Okay. Again, make sure that chips are sterile. Next one is scope. Placing it sometimes can be challenging, as you can see. And then next, some microquat. Okay, let's grab a paper tip. Dip in the microquat. Okay, here we go, microquat. More to go. Lysol. And finally, okay, ethanol is gone. Let me see what I can do. Alright, you managed to recover ethanol. So that is our last disc. Piece of paper, and done. So I'm not gonna show you same procedure for salmonella, but staphylococci are done. So you may see the blue light on the side of a picture. Side of the screen. It's the UV light working. So what I'm going to do, I'm going to expose half of these plates to the UV light, and half of them will be left unexposed. So I'm going to separate it like this. I'm going to put nice UV note. Okay. And then we're going to cover one half of the plates with this makeshift covers, and we're going to put this plate under the UV light for exactly 1 minute, and I'm going to try to show you what it looks like. Okay, so they're sitting under the UV light. They're going to stay there for a minute. Minute expired, turning off the light. So now I'm going to put these plates in the incubator, and well, we will see what we get out of it in a couple of days.